Thanks so much, Nick. My name is Omar Ahmed. I'm an associate professor at the University of Michigan. And this movie came out probably 19 years ago, I think 2004. I probably saw it 16 or 17 years ago, sitting in my apartment in Providence, Rhode Island, probably on my brown couch. That's what I'm trying to pretend I remember right now. And I'm pretty sure I enjoyed it. I'm pretty sure I loved it. How could I not? It's about memories, and that's what my lab studies. That's what I study. That's what I've always loved. But I can't tell you that for sure. I think I saw it on a DVD, using a DVD. I think I got it from Netflix when they shipped DVDs. Could have been from Blockbuster. Yes, it's that old memory that Blockbuster was still around, for those of you who remember that. So, yeah, some pretty ancient memories that I've probably edited and reinforced as we speak. And I guess today, we'll just kind of talk about where in the brain are memories stored and how real a phenomenon was discussed in this movie. Can we actually edit memories? How close are we to doing that? So to answer the question of where memories are stored, let's actually start by discussing epilepsy for just a minute. And it'll become clear why epilepsy. So 50 million people or more suffer from epilepsy. That means that they have more than one seizure each. And it was first described in the 2nd century AD by Aristeas of Cappadocia, an ancient Greek physician. He also actually was the first person to describe diabetes. And he called it an illness of various shapes and horrible. And he recognized that it took on various shapes. It could manifest itself in many, many different ways. And now, for that reason, it's often referred to as the epilepsies. The reason I bring that up is because probably the most famous person and patient in the whole field of neuroscience, named HM, had temporal lobe epilepsy. That's one of the more common kinds of epilepsy. And HM's epilepsy could not be treated with drugs. They tried many of them for 20 years. HM had suffered from seizures. They were debilitating. And eventually it got to the stage where his neurosurgeon decided, let's remove the parts of HM's brain that are probably causing his seizures. And they proceeded to remove both his left and his right hippocampus. And I think many of you have probably heard this story before, but so what happened after HM's neurosurgeon went in there and said, we're going to free you of your epilepsy, we're going to remove this part of the brain called the hippocampus, and you're going to be better. So what happened is that HM's epilepsy was now under control. So good news, right? But HM was not really the same person anymore. He suffered from debilitating amnesia. He could not remember people he had met a few minutes ago. Experimenters would come into the room, introduce themselves again and again and again, pretty much every single day, every few minutes they would have to reintroduce themselves. He underestimated his own age. His memory had kind of stopped. He had stopped aging at the point at which his hippocampus was removed. Everything he remembered was at least from before the time of his surgery, and sometimes people think maybe many years before his surgery. And he described the state as like waking from a dream every day is alone in itself. And I think you can probably appreciate how debilitating and disorienting that must have felt every single day. So HM passed away in 2008 at the age of 82. His full name was revealed, Henry Molaison. People spent 60 plus years studying HM, working with HM, trying to understand his brain, trying to understand memory and how it works, because HM did not have a hippocampus in either side of his brain. And that's how we really figured out, look, the hippocampus is critical for forming new memories. Now we also know that the hippocampus fails early on in many neurological disorders that involve memory impairments, chief among which, and perhaps best known, is Alzheimer's disease. And Unsurprisingly, people with Alzheimer's disease show progressive memory loss. Here's one example. So this is a case study of another patient named MD. And MD's behavior, in this quote, clearly reflected her episodic memory deficit, similar to an amnesic patient, so like HM. She never recognized the experimenter, but was always eager to participate in the sessions. So very similar to HM, but MD is suffering from Alzheimer's disease. The other thing, though, that comes with Alzheimer's disease is in the quote that appears just below. So MD was a very sociable person. She used to go to community church 2 or 3 times a week. Her husband needed to escort her to the game and rosary activities, which were virtually her only remaining hobbies. However, her husband couldn't take her every day. So when he wasn't there, she attempted to go on her own and got completely lost. And after that, he was worried about her, so she stayed home. That aspect of Alzheimer's disease, that people start to get lost, they become disoriented in space, they become disoriented in time. That is another essential element of what these parts of the brain that encode memories, they're critical for actually giving us way more than just a memory here or there. They actually define our personalities. They help us navigate our worlds in both space and time. Here's another example. In 2002, this other patient recalled that on several occasions he got lost while walking in familiar streets, even though he was absolutely aware of where he was going. He's on State Street, he wants to get to Main Street, he knows that, but he can't figure them out. He can't connect the dots, he can't figure out which way to go. 2 years later, his Alzheimer's got worse, and at that stage, the patient had developed difficulties even in moving from one room to another in his own house. The patient complained that he had to proceed by trial and error, going from room to room in his own house to figure out, this is where I wanted to go, I'm here. It's that profound. And it's now recognized that this spatial disorientation, but also more broadly disorientation of many different kinds, is a very common symptom in Alzheimer's disease. 93% of patients with Alzheimer's disease suffer from spatial disorientation. But it goes further. It's not just Alzheimer's. Almost all people with Parkinson's disease also suffer from this kind of spatial disorientation, coupled with some memory loss as well. This quote kind of explains why it's underdiagnosed in people with Parkinson's. I used to walk alone in the woods, fog or no fog. But when the symptoms of Parkinson's appeared, I realized I would easily get lost, especially in the case of fog. Now I am too disoriented to get lost anymore. So because Parkinson's disease comes with these motor symptoms that prevent people from moving around and navigating their environment seamlessly, that's why it's underdiagnosed. They can't actually even navigate enough to see that they are spatially disoriented. So here's one more example. He was referred to our department because 2 weeks ago, while driving his car, he suddenly realized he was unable to recognize the streets along his way home. He got lost and had to call on passersby to help. Since then, the patient has been severely disoriented in space. So, any guesses, Alzheimer's or Parkinson's? All those in favor of Alzheimer's, just raise your hands. So, Parkinson's. This one's actually neither. So, in this case, the person had actually had a cancer, a glioblastoma, around a part of the brain called the retrosplenial cortex, not the hippocampus. It's a part of the brain called the retrosplenial cortex, and recent work has shown that this part of the brain is also critical for memory and for spatial orientation, and is one of the first to go in Alzheimer's disease and probably Parkinson's, although we don't know that for sure. Here's a drawing done by a person with a lesion of their retrosplenial cortex on the left, and their son drew the same neighborhood of theirs on the right. You can appreciate that the patient actually left out many of the details, many of the buildings, many of the landmarks are missing, but also many of the angles connecting certain landmarks are wrong. Memory and space, they fade together, and I think this kind of essential component that we are embedded in our spatial and temporal world. Space and time is everything. They are the scaffold around which every single memory we form is built. And without certain brain regions that are so essential for space and time, our memories can also start to fade. Here's one last case study. So another person with retrosplenial damage. His retrograde amnesia increased post-operatively to encompass more than 4 years. He did not remember the birth of his second child, who's now 4 years of age, believing he had only one child. His anterograde amnesia, meaning the inability to form new memories, was profound. By lunch, he could not reliably recall what he had done that morning. This quote could have been from HM, or could have been about HM, but it's about a person who had a damage to a completely different brain region. And I mention all this to say that the hippocampus is what we always think about as being critical to memories, to kind of forming all of the associations that make us who we are. But it doesn't work alone. It works with many other brain regions, like the retrosplenial cortex, and regions like the hippocampus and retrosplenial cortex are both altered, damaged in different ways early on in people who will go on to develop Alzheimer's disease and other neurological disorders. So how does the recall of memories work, and why is it so subjective? So here's a little experiment. So fruit A, fill in the blank, something that starts with A. Apple. I mostly heard apples. I had one apricot. I heard one more. What's that? Avocado. Excellent. It is a fruit. So here's an apple and an apricot. And I've been trying to train myself actually to stop using the British way of saying it, like apricot, as opposed to apricot. So right now I'm stuck somewhere in the middle. So if my pronunciation of apricot or apricot sounds weird, please forgive me. So here's fruit A. You hear fruit A. Your brain, your hippocampus, your other brain regions have these little gravitational wells. When you hear something, you want to put that information into one of these wells, and that'll lead you to complete your memory, to recall something interesting. So most of you, when you hear fruit A, you have a gigantic well that corresponds to fruit A must mean apple. And that happens, right? And you think apple. Now all you can think about are apples. That's taken over your brain. You know that fruit A should lead to apple. But when I asked this question in a class a couple of years ago, another student had said apricot. And I asked her, why did you say that? Did you just see an apricot? Did you just have one? And she said, yeah, I had one for breakfast. Recency effect. It basically widens that gravitational well for apricot in this case. And in her case, fruit A falls into that particular well. It's very easy to change what we're going to recall based on our current surroundings, based on what we did recently, based on where we are, based on what we're feeling, based on what we're sensing right now. Similarly, post-traumatic stress disorder and other, perhaps not PTSD-like, but strong emotional experiences can severely impact memory and how we react to certain things. So for example, if someone's been in a car accident that involved a lot of loud noises, or someone coming back from the battlefield where they'd heard lots of explosives and had many scarring experiences there, if they now, in a fairly forgiving setting, hear a really loud noise, that might often elicit a fearful reaction because the well, or the gravitational well that is formed associating that sound with fear is just huge. And so one of the things that people try to do to actually treat PTSD-like symptoms is something called retrieval-induced forgetting. You can try to use the retrieval of certain memories, perhaps associate them with slightly different things. And what you're really trying to do there, this big, gigantic gravitational well associated with loud sounds and fear, you're trying to just shrink it down so that the other wells can take over and replace it over time. It's not quite what's happening in the movie, but you can see similarities there. So Memories depend on context. That really is the point here. Our past, present emotions, current location, current sensations, all of this can influence what and how we remember at any given moment in time. I really liked in the movie where Joel was trying to take Clementine into these really different spaces, these really different childhood times periods, so that he could associate her with that space and time, far away from where they were looking, far away from the memories they were trying to eradicate. But in the actual brain, there are no gravitational wells. So how is this all done? Well, the human brain has 86 billion neurons, all working relatively in synchrony with each other. And 50 to 100 million of those can be found in the human hippocampus, which is this little structure right here. It looks like a ram's horn or perhaps like a seahorse. And hippocampus actually comes from Greek. Hippos is horse, campus is sea monster. Campus also means bent, like a bent horse, like a seahorse. So we're not quite sure what the origin of the name referred to, but it's either seahorse or a mythical creature that is a cross between a horse and a mermaid, supposedly. But either way, it reminded a lot of people of seahorses. And this is what the rodent hippocampus in rat and mice kind of looks like. And in rat and mice, you can put electrodes in and study neurons in the hippocampus and see what they're doing. When you do this, and then you take a little rat or a mouse and they run in a straight line, it's basically same things happening in my brain right now. I'm just walking across the stage here. In one location, right here, one neuron will start firing. By the time I get over here, it's stopped. Some other neurons are firing, and another neuron, and another neuron. So one neuron fires in one given tiny location. That neuron is called a place cell because it encodes a region of space, a place, and it has its own place field. Place cells, just like that one, are seen in humans. Going back to epilepsy patients, when epilepsy patients get implanted, you try to record from their brains to figure out where their seizures are coming from. And with these electrodes in their brains, you can actually record neurons in the human hippocampus. And you can have them play virtual reality games, like a taxi driver game, while they're sitting in their hospital bed. And when these kind of experiments are done, people have found that neurons in the human brain, in the human hippocampus, that are basically place cells, even in a virtual reality taxi game. In this case, the bright red square is basically where the neuron will fire most every single time the person drives their taxi through that square. Probably every place you've ever visited has a group of place cells encoding it. So 100 million neurons, different combinations of them can encode different parts of your world all the time. Right now, 50 neurons, another 50 neurons, another 50 neurons, another 50 neurons. Every single part of space is tiled over by these place cells. Why? Right? Well, space is fundamental, right? We evolved to kind of navigate our space. Mammals need to leave their houses, go hunting for food, find shelter. You need to navigate your space. All other memories somehow come along with that. Everything is embedded in trying to navigate our space. And hippocampal place cells, so is it just space? Well, no. So blind rats have place cells, so it's not just vision that is causing it. Anosmic rats, rats that cannot smell, have place cells. Deaf rats will have place cells. So it's a lot more than just vision. But even if you take a rat or a mouse and have them run in a hamster wheel, so it's kind of like me just standing here and then just me walking in space. Is my location changing? No. But a sequence of place cells, A, B, C, D, E, will start firing as I start running in my hamster wheel, or as a rat starts running in a hamster wheel. So it's not even place. I'm not changing my place, but I have different place cells firing because I'm walking or I'm thinking about something. Place cells are a lot more than just place. They're also time. They encode objects. they encode people, they encode everything. They basically are the correlates of memory. Sequences of place cells is what get activated when we remember anything. When we encode a memory, we encode it with sequences of place cells. And even though the first place cell was seen in 1971, it took of them a few years, but the 2014 Nobel Prize was awarded to the discovery of place cells. So they are a fundamental discovery that is the unit of memory and how it's encoded in mammalian brains. So just to kind of wrap up and kind of tie this back to the eternal sunshine of the spotless mind. So are we ready to alter specific memories with the kind of surgical precision they kind of showed in the movie? The short answer, unfortunately, is no. But recent work in mice and rats kind of gives us some clues about a way forward. So this is the same figure we saw a few minutes ago. All these groups of place cells encoding different locations in space. As you can see, the dark blue place cells right at the end there, they're closest to the cheese, right? They're closest to the reward location where the rat or mouse goes and stops and consumes its reward. What if you found a way to identify those dark blue cells and activate them, not at the end, but in the middle of the track, when the rat is running down this track, and instead of at the end, they get activated right here. What do you think the animal, the mouse or the rat would do? It would get the cheese. Did I hear that correctly? It would look for cheese. It would stop and look for cheese. Exactly. And that's pretty much what happened when they did this experiment. Zap the dark blue cells, stop. Stop and look for cheese. Why? Because the cells that encode the cheese location are firing, so I must be at the cheese location. It's time for me to eat cheese. Right? Memories can be manipulated. This is manipulating a memory. It is possible. It all comes down to a bunch of cells, each one encoding different aspects of your environment, different features, different people, different memories. If you can activate them at the right time, you can induce a so-called false memory. You can induce a modified memory. It is possible. The equipment to do an experiment like this looks something like that. It's a gigantic microscope surrounded by really bulky, expensive, heavy-duty, powerful lasers that let you see neurons in the rodent brain and then zap them as well to change their activity patterns in real time. And this technique is called 3D holography. That's what it kind of looks like. You can basically zap any part of the brain with 1 cell at a time or 10 cells at a time. As you can probably imagine, none of this is going to make it to humans anytime soon. But in a sense, what did they have in the movie, right? They had this kind of fMRI-like device sitting around Joel's head while they manipulated his memories. So if we can figure out ways to non-invasively activate single neurons, it's a pipe dream right now, or groups of neurons, and to record their activity, one day that may be possible. So just to want to thank Nick, the Michigan Theater, and the Science on Screen series for giving me the chance to talk to you all today, and happy to take questions. Thank you.
Yes. Thank you, Dr. Ahmed. Yeah, like I said, we're going to take questions now. I will open up the floor to questions. With first my own, I'll give you a chance to think of yours. And so, if, as you described, or maybe correct me if I'm wrong, so as where I'm staying right now, these place cells are firing where I am right now. Say, if I went home and I dreamed about this tonight, are those neurons, are those the same neurons that are refiring? How does that work?
That's a great question, and the answer is exactly yes.
Okay.
So let's say, I'll simplify it. Let's say I walk down A, B, C, D, E, and I'm just going to take a nap right here. All right, I'm going to fall asleep right now. When I fall asleep, my brain's going to replay A, B, C, D, E, A, B, C, D, E, A, B, C, D, E. especially if I got a reward or something meaningful happened to me right here. So, this is my cheese. A, B, C, D, E led to cheese, fall asleep, dream of cheese, replay A, B, C, D, E. Strengthen that memory. Pass it from the hippocampus to the retrospinal cortex. Store it for longer. Recall it the next day. Modify it later. So absolutely, when you fall asleep, your experiences get replayed. The place cells literally get replayed. And they are the basis of that memory, and they help to strengthen that memory while you sleep.
Raise your hand if you have a question. I'm gonna run them. Yeah, I'll do my best to get to as many questions as I can, but I'm physically running around with the microphone, so you're here.
Thank you. I have a question about, do place cells have anything to do with the feeling of deja vu?
Yeah, I think they absolutely can. So this is not quite deja vu, but it's close. But imagine this A, B, C, D, E. Let's say somehow you manage to fire off A and B in some other context, in some other location. If neurons A and B fire, they might actually lead to C, D, E because you're so used to thinking about that sequence. And that can complete a memory or kind of recreate a memory or give you this feeling that I've seen all of this before, even though it's not exactly the same. So yeah, they would almost certainly have to be involved in that kind of percept.
Hi, I was wondering how the lack of ability for humans to manipulate their selective memory might have aided us in getting this far in terms of evolution, whether that be family structure or retaining traumas to avoid future ones. I was curious your opinion on that.
Yeah, so clearly we remember things that are negative in some ways, that induce fear, that cause us to be careful, that cause us to avoid certain places, that cause us to say, I'm not going to touch that hot iron because that's just dangerous. And especially from an evolutionary perspective, I think you're absolutely right. Because why are there so many ways for our brains to associate things with fear? Why are there so many ways for our brains to associate things with freezing and running away? Well, because it served, exactly as you're saying, a massive evolutionary function. It helped animals survive. If I'm a mouse and I see a looming shadow, something's about to swoop down on me. I'm going to freeze. I'm going to hide. I'm going to be afraid. Why? This will help me survive. And this is why those circuits, those pathways remain embedded in our brains. And yes, they serve us. They serve a function. They're supposed to help us. But sometimes that wiring can actually hurt us.
So kind of an odd and layered question, but background slightly, the past couple of years, I've lived in a couple relatively traumatic situations and lived in my car for like a little over a year, and I noticed the neurochemical ability to be creative and recall memories has effectively gone in a lot of ways, and I think through a flood of norepinephrine, a lot of any capability for neuroplasticity severely lessened, and sometimes, honestly, through aid of like THC and some things, I'm able to sort of dive back in. But I wonder, does the brain have a system in which it sort of creates a scar tissue, as in if there are parts of the brain that other parts of the brain are attempting to hide, sort of. Like in this, they're sort of erasing that. If there are parts of the brain that are attempting to hide, sorry, I'm trying to phrase this.
That's okay.
Is there a threshold at which it can become harder to access those memories? Does the brain sort of attempt to mitigate even further any access to those memories? Or is one able to push through at the same level regardless of experience?
Yeah, I think just more broadly speaking, rehearsal experiences, revisiting places, revisiting thoughts, revisiting memories, they tend to keep those memories and related memories alive. I kind of, and if those experiences haven't been happening for a while, then it's harder and harder to access the memory of those experiences. In my kind of really rather basic example of that I started with, like, I was racking my brain trying to remember the experience of watching this movie for the first time. And I know it's there. I know I enjoyed it. I can't remember the details. I didn't remember most of the details of the movie. I have this vague notion that the movie was a lot of fun because it must have been, right? How could it not have been? But where is that memory? Why can't I remember the instant of watching that movie? Because I haven't really thought about it for a really long time. I haven't rehearsed it. So yes, I mean, it's not so much that the region of the brain disappears, but the group of neurons that encoded that memory haven't been reactivated together for so long that they can become hard to access. That said, it's in there. There was this is a slightly unrelated anecdote, but when an epilepsy patient gets implanted with electrodes, you're trying to stimulate those electrodes as well to see if it induces a seizure. And so the clinicians do that. And I was in the room once when they're doing that in this patient and just making sure that this is the part of the brain that can elicit a seizure. And they stimulated a part of this person's brain, and it didn't elicit a seizure, but his eyes kind of glazed over, and he just kind of looked off in wonder. And when the stimulation ended, they asked him, what just happened? And he said, well, I just remembered something I haven't thought of. You know, he was probably in his 40s. He said, I just remembered being in 4th grade, sitting with my friends, reciting the Pledge of Allegiance. 4th grade, you know, so a memory that he had not thought of in 30 plus years. So it's in there. Perhaps what's really changing is the way that we can access it, that pathway that guides us, that guides our brain to access that memory is what's kind of degraded, and finding that pathway is really the challenge.
Any other questions? Raise your hands. And I'll run my... Yeah, I'm going to go over here, and then I'm going to make my way back over there.
Hi, I noticed one thing. It's common after people, say, hit their head in a car accident, happened to a friend of mine, that they can't remember for some number of, it's usually like hours before, you know, they're like, a friend of mine was in a severe car accident, and like at noon, and she said, you know, I remember getting ready for work that morning, and then it's gone. Is there some sort of short-term, like time that it takes to encode memories and longer term, or what's going on there?
Yeah, absolutely. It's a great point. So our recent memories need to be replayed often, perhaps in the next night's sleep or the next night's sleep, etc. So in the case of a car accident and the trauma associated with it, the physical trauma that the neurons could suffer, but also just not getting sleep, kind of just being overwhelmed with these neurotransmitters bathing the hippocampus, and the hippocampus not being able to do its normal function during your subsequent sleep. So that can explain why some of the memories that you should have formed and did experience just do not get consolidated. The corollary of that is that in patients like HM, where you remove part of the hippocampus, or where the person had a retrospinal lesion, in both of those cases, patients were unable to form new memories, but just like you described, they also didn't remember some of their recent memories. In the case of the retrospinal damage, the last 4 years. Where did they go? And why the last 4 years? Why not the last 40 or the last 20? Every time we go to fall asleep, some of these memories, they get consolidated out of the hippocampus and to the rest of our brain, to places like the retrospinal cortex and other parts of the cortex. There's constantly being moved and shuffled further and further away, if you will, from the hippocampus, and stored away for recall in the future. So when you remove HM's hippocampus and nearby regions, that removes some of those more recent memories, but spares the really older memories. So yes, traumatic events or removing parts of the brain can lead to forgetting about recent experiences.
So I learned recently about a new study where they used psilocybin from magic mushrooms just to see how it affects your brain. And since it's new, it's not super legal yet. a lot of the participants described it sort of like having a dream, but like you're awake. Is there a connection between what drugs like this do to our brains and what happens when we're actually dreaming? And then how can that be used to kind of either like innovate or, you know, make an argument for them?
I think there's a definite connection. Any chemical can alter how we recall and understand and form memories. So actually, a different part of my lab is studying psychedelics and how they influence all kinds of neural circuits. And what psychedelics can do, and this seems to be one of the things that they do very reliably, they boost the number of synaptic connections from neuron to neuron, which might sound like a very useful thing, and I think it is, right? So if you could boost the connections between neuron A and B and C and C and D, that sounds like you could then remember A, B, C, D, E, and it led to cheese in my rat-based example from earlier. So in that sense, it could be very positive. More synapses, good thing. On the flip side, what we don't know yet and we need to understand in the case of psychedelic drugs is, are they boosting all of these synapses? What about A to E? What about E to A? What about A to Q, right? All kinds of other connections could also be made. Perhaps they're useful, but perhaps they may actually lead to some kind of confusion about recalling that memory. And I think that's what we still need to understand. More synapses is good, but if they're not specific, are they still good? And I don't think we know the answer yet.
You kind of just touched on this, but people with super memories, what's going on there?
I wish I knew. No, I mean, it's...
He doesn't remember.
Yeah, it's, you know, so in childhood, there's this like critical period where you can form all kinds of memories. Your brain's really plastic. You can really wire up the brain in any way you see fit. And some people think that psychedelic drugs could even be opening up that critical period again. Perhaps in people with super memories, that critical period is just a little bit more open their entire lives. Right? It kind of never fully shuts. You can just constantly keep encoding this new information better than most other people can who are trying to retain the things that they have learned. But really, super memories is something that I don't think have been cracked in terms of why. There's some molecular pathway, and those pathways could be different from person to person, from super memory person to super memory person, that allows them to just remember things better and recall them better. But also, on the flip side, some people with super memories can suffer from almost being overburdened by these kind of recall attempts. So, you know, kind of limiting that critical period might serve an important purpose as well.
Yes, you brought up Alzheimer's in your presentation. My mom had that. And have they made any progress in the last 5 years?
There are some new drugs that actually try to use almost, there's a byproduct, a protein amyloid beta that is just not very good, and it correlates to progressive stages of Alzheimer's disease. So many pharmaceutical companies are right now trying to design ways to actually take this away, essentially like antibody-like treatments that bind to the amyloid beta and remove it. A couple of recent drugs have shown promise, but I think that's the best that can be said right now. They need to be evaluated for side effects. They have shown in a very recent study that over the course of, I believe it was 18 months, a group of patients treated with one of these new drugs didn't worsen as much as a group of people treated with placebo. So, you know, that's not saying a great thing. They didn't get as bad as someone who was treated with placebo, but they also still got worse in terms of their memory capabilities. So there's promise ahead, but I think a lot more work needs to be done on these kinds of drugs. But there have been some breakthroughs recently.
I'm going to take a question that I promise we'll have time for about 2 or 3 more, but my question is related to this. Even if Alzheimer's is able to be reversed, the memories and the details that were lost, do you think that they could ever return, or are they gone forever?
Yeah, so the thing I think in Alzheimer's is that those memories are still there. I mean, my grandmother had Alzheimer's too, and my mom always told me the story that, she wouldn't remember, my grandmother wouldn't remember my mom for 95% of the time, but there would be these amazingly lucid periods where she knows everything about her, and and I think anyone who has family members who've had Alzheimer's know that that's possible. It's in there. It's still in there. It's much like the person remembering the Pledge of Allegiance from 4th grade. The memory is still in there. What Alzheimer's seems to involve is that there are all these other regulatory pathways in the brain, something called the cholinergic system that kind of controls the entire brain. Those regulatory pathways don't quite do their job, so that when it's time to remember a memory and do it precisely, that's what fails. The memory's still there. You can't pull it out at the right time in the right context. So yes, if you fix the regulatory pathway issues, many of the memories may still be waiting there, ready to be recalled.
I had a photo regarding, or I had a question regarding the photos in the film. Clementine and Joel both wanted to get rid of photos of each other to sort of avoid that memory recall. I was curious, if I were to see a photo of a place that I've been and it looks different than how I remembered it, would that photo alter my memory then and sort of change the way I recall it going forward, or are those effectively always separated?
Yeah, great question. So you've been to Bermuda, let's say, and you remember a particular beach, and now you're looking at a photo of that beach and it looks slightly different, and perhaps you're not even sure whether the photo's the one that's different or if your memory itself is not quite right. Right? So, and suddenly now, which one is it? And I think that's also similar to me making up or completing this old memory about watching this movie 17 years ago. Whenever we re-experience a particular thing, the very memory of the initial object, the initial photo, the initial beach, the initial watching of a movie, it has to get modified. That's just how the brain works. It's not like a computer where it's just stored in a little bit of RAM and that's it. It'll always be identical. Every time we recall and think about or re-experience something related, the initial memory can get slightly modified, and that's why they are subjective. That's why they are probabilistic. So short answer, yes, I think that'll change your initial memory a little bit. And I think we all then have to work very, very hard to kind of hold on to what is real. And I think, that especially kind of when inundated with information, that's a really difficult challenge.
Do we have one more question? I'm gonna come way back here.
All right, my question would be like, I know you touched on things like neuroplasticity. Would like the neurons in the brain be like structured statically or consistently enough to where you'd be able to like assign like addresses to each, like in a manner like that the movie alludes to, like in the computer's interaction with the brain?
Yeah, probably not, and here's why. So let's say right here, I've got neurons A, B, and C encoding this location, and I remember that A, B, and C fire, and I remember that you asked me that question, kind of relating computers and brains and how memories are stored and addressed. I'm going to move over here now. It's very possible that neurons A, Q, and Z are now firing. A is involved in both locations. What I'm trying to say is that the same neuron can participate in encoding many different memories. It's the group of neurons together that allows you to recall the specific memory. Why is this important? Because you only have, as I said, 50 to 100 million hippocampal neurons. Sounds like a lot, but it's not infinite. You don't want to be limited to only 50 million memories, right? I mean, you've probably walked way more than 50 million centimeters. And if you're only limited to 50 million centimeters worth of encoding, that's not enough. Whereas by using neurons, groups of neurons in combination to encode different kinds of memories, you exponentially increase your capability for memory. So short answer, I think it's very different from the precise address that a computer would use. In this case, it's a probabilistic combination of multiple neurons encoding every little precise memory.
Thank you.
Yeah. I think we have time for one more question. I know I said that was the last. I'm going to come over here, and you are going to have the last.
So I'm curious about, you recall things that happened to you when you were like 2 because it was traumatic. So at least to a 2-year-old, it's traumatic. But then your day-to-day mundane things, you can't recall them at all. So is it that trauma that, I guess fight or flight, it comes back to you when you feel like you're in a similar situation? Or is there something else that's going on that you can remember those things, even if you're not in that same situation?
Yeah, it's a great point. So I like thinking of memories as these gravitational wells for exactly that kind of reason that you're bringing up, which is why are some memory wells bigger than others, right? And I think if we take the evolution of the brain and the importance of avoiding danger, hence for better or for worse, the importance of fear, we can imagine why there are lots of wiring diagrams in the brain that say like, well, if something's traumatic, let's really overemphasize that. Let's really encode that way more than perhaps we ideally should. Let's represent it with a huge well that essentially means it's really easy to access that memory. And that's why certain memories, no matter how old, could be easier to recall because this gravitational well for that memory is just huge. And for better or for worse, often fearful memories, traumatic memories can lead to larger gravitational memory wells, allowing you to recall that all the time in the future. And again, that served an evolutionary perspective in many other species, and perhaps to some extent in us as well, but it now gets usurped and doesn't function as we would like it to.
Well, thank you so much, Dr. Ahmed, for coming out. Before we leave, where can people go to find more about your work?
So omarlab.org, omarlab.org is our lab website. Some of our papers are there. My contact information is there. And you're welcome to start there.
And I'm going to put you on the spot right now. What is another film that you recommend that we should go home and watch tonight and also related to your work as equally as...
Can it be a TV show as opposed to a film? Severance.
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 All right.
It is related and not related in ways that I just love to this movie as well. So yeah, it's totally worth watching.
Terrific. Please help me give another thank you to Dr. Ahmed.
Thank you.